concentration of substance in just seconds, or how they identify unknown compounds without touching them. The secret lies in the one of the most powerful yet elegant instrument in chemistry, the UV visible spectrophotometer. Welcome to Science Simplified. In the next 10 minutes, I am going to break down everything you need to know about UV visible spectroscopy, from the fundamental physics to practical applications. Stick around, because by the end of this video, you will understand exactly how this incredible technology works and why it's used in everything from crime labs to pharmaceutical companies. And for those of watching for exam preparation or lab work, I have prepared this with all the key formulas and practical tips you need. Let's dive in. What is UV visible spectroscopy? It's all about light absorption. Coming to the definition, UV visible spectroscopy is the study of absorption of UV light and visible light by molecules. When light passes through a sample, some wavelengths are absorbed, and this absorption gives us information about concentration, structure, electronic transitions. About basic principles, the core principle is beautifully simple. When light of the right energy hits the molecule, it can excite electrons to higher energy states. Different molecule absorbs different wavelengths based on their electronic structure. Every compound has its own light fingerprint, specific wavelength it likes to absorb. Our job is to read that fingerprint. These are common fingerprint transitions: sigma to sigma star, n to sigma star, pi to pi star, n to pi star. What is this? This means compounds containing double bonds, aromatic rings, and lone pairs of electrons shows UV visible absorption. These are the transitions related to this double bond, rings, and lone pairs. Coming back to the law, the heart of all its, the Beer-Lambert's law. Now for the mathematical magic behind it all. The Beer-Lambert's law. This is the equation you need to know. The formula is A equals to epsilon into C into L, which means A stands for absorbance, means what we measure. Epsilon is the molar absorptivity, means a constant unique to each compound. C is concentration, what we often want to find, and I is the path length, usually 1 to 2 centimeter, or usually 1. Here is the key insight. Absorbance is directly proportional to the concentration. Double the concentration, double the absorbance. This linear relationship is why UV visible is so powerful for quantitative analysis. Coming back to instrumentation, let's take a virtual tour inside the instrumentation. Our instrument, every UV visible spectrometer has 5 components. The highlights of the components. First one is the light source, usually 2 lamps, a deuterium lamp for UV light and tungsten lamp for visible light. And second one is the monochromator, the wavelength selector. What is the work? It uses a grating or prism to split light into individual wavelengths, so we can test them one by one. The third one is the sample compartment, where the magic happens. This holds your sample in a separate transparent container called a cuvette. Pro tips: Use quad quads for UV work. Glass blocks UV light. Fourth one is the detector. Typically a photomultiplier tube that converts light intensity into electrical signals. Photodiode, second one is. And fifth one is the computer and software, which process all this data into beautiful spectra and calculations. The entire process happens in seconds, giving us a precise, reproducible results. Types of UV visible spectrophotometer. Two types: single beam instrument and double beam instrument. For comes to the single beam, the measures blackened sample separately, and it is less expensive and less accurate. For the double beam instrument, they splits light into two beams, measures blackened sample simultaneously, and it is more accurate and widely used. Coming to the calibration and system suitability, it plays very important role for the instrument. USP strongly emphasizes instrument calibration. For the parameters we are checking for the calibration: wavelength accuracy using holmium oxide. For photometric accuracy, using potassium dichromate solution. For the stray light test means an unwanted light reach it to the detector without deviating the process. It ensures light does not reach to the detector. And applications: UV visible spectroscopy is used in many areas like pharmaceutical industries, checking drug purity and concentration. In environmental science, measuring pollutants in water. In forensic, identifying unknown substances at crime scenes. In biochemistry, studying protein DNA interactions. Pro tips and common pitfalls. Before you run to the lab, here are crucial tips from year of experience. First one is always run a blank first. This corrects for solvent absorption because it minimizes background or noise. The second one is keep absorbance readings between 0.1 to 1.0 for the best accuracy. Too low the concentration, concentrate your sample. Too high, dilute it. And the third one is beware of the stray light effect at every high absorbance. Fourth one is clean quad properly. Fingerprint scatter light. And the most importantly, never look directly into the beam. UV light can damage your eye permanently. Follow these and you will avoid 90% of common mistakes. Thank you for watching my videos. If you like the video, hit the like button and subscribe the channel.